Hello everyone, welcome back to the session. So in today's class we are going to discuss about the very short answer question for structural organization animals as well as digestion and absorption. Okay, sorry, breathing and exchange of gases. So we have started the very short answer questions and we have completed two chapters in yesterday's class, and in today's class also we are going to complete two chapters. We'll start, okay, and this particular chapter is structural organization animals in the first year. Okay, so the question: the radial symmetry is an advantage to sessile or slow moving organism. First you should know what is meant by radial symmetry. Okay, so what is meant by radial symmetry? See, consider this as an organism. Okay, so this is an organism, and if we cut a particular organism at any plane, okay, we can come up with two morphologically similar equal halves. Okay, so that is what radial symmetry. Okay, so sessile means non-moving. Okay, it's sedentary. Okay, non-moving. Slow moving means it will be a slow moving organisms. Okay, see here. See, consider this for the sessile animals or the slow moving organisms, the radial symmetry is considered as an advantage. Why? See, animals with radial symmetry will live in water, no doubt in that it will be living in the water. Okay, so they can respond equally to stimuli from all directions. So this is important. See, those organisms which is present in the water, they can receive stimulus equally from all directions. Okay, which means those organisms, which means those stimuli which is coming from each direction, they can respond equally to all stimulus coming from all the directions. So that is why this radial symmetry benefits the sessile as well as the slow moving animal. So this is the proper answer for this question. See here, so for this question you have to answer the radial symmetry for which is living in the water. Okay, so as it is we are living in water, so they have to respond equally to all the stimuli from all the directions. Okay, this is the main important reason for this question. Okay, so hence radial symmetry benefits the sessile or the slow moving animals. Okay, so this is the first question, and the second question will be, what is cephalization and how it is useful to its possessors? Okay, see, the cephalization is means cephalo. The word cephalo means head. So the formation of head is nothing but a cephalization. Okay. So this cephalization process, how it is useful to its possessors. Possessor means those who are having a head portion. Okay. See here, the concentration of nerve and sensory cells at the anterior end. Anterior end means the head. The anterior end, and the posterior end means tail region. The head region is anterior and the tail region is posterior. Okay. Now at the anterior end, the concentration of nerve and sense cells are higher if there will be the formation of head region. Okay. Now this is the one enables the animals to sense and responds to newer environments efficiently and quickly only whenever there will be the formation of an anterior region. See here, the cephalization, the word cephalo means the head region. Okay. What if the head region is formed? There will be the formation of sensory cells as well as the nerve cells. We know how the nerve cells and sensory cells are very important to respond to the stimuli. Okay, so that is what, as the concentration of nerve cells and sensory cells are higher in the anterior end, so this enables the animals to sense as well as to respond to the newer environment efficiently and quickly. This is the important keyword. Okay, so this is why the cephalization is useful for its possessors. Okay, now the third question will be, animals that exhibited tube within a tube organization first and their body cavity. See, animals exhibiting the tube within a tube. Okay, tube within a tube organization first and their body cavity. So you should know the body cavity will be present in. So the body cavity will be absent up to the, we are having a phylum Porifera, Nidalia, Ctenophora, Platyhelminthes. Okay, Platyhelminthes. All these phylums are not having the proper body cavity. Okay, and the pseudocoelom, which means the false body cavity, the concept of body cavity will be started to arise from the nematodes, that is the phylum as helminthus. Okay, that is the round worm. So those are all the worms which are having a body cavity like structure, but actually it is not a proper body cavity. Okay, so the concept of body cavity has been originated, even though it is not a proper body cavity, the concept has been arise from the phylum nematodes. Okay, so from the phylum annelids, they are having a proper body cavity. We can call them as the eucoelom. It's EU. Okay, eucoelom. Okay, so that is what the proper answer for this question. Okay, so the fourth question will be, why is the true coelom considered as a secondary body cavity? See here, so here in this question, so this is what a tube within a tube organization, that is a pseudocoelom. But here the true coelom is considered as a secondary body cavity. Okay, so this is an eucoelomate, which means a true body cavity. The blastocoel is replaced by a true coelom, which is derived from the mesoderm. Okay, so hence it is called as a secondary body cavity. Okay, see here, eucoelomates means those who are having a true body cavity. We are having acoelomate, pseudocoelomate and eucoelomate. So this pseudocoelom is considered as a primary body cavity, that is the tube within a tube. Okay, so this eucoelomate is a proper body cavity and it is called as a secondary body cavity. Here in eucoelomates, the blastocoel is replaced by a true coelom, which is derived by the middle layer mesoderm. So you should know that if there is a true body cavity, that cavity will be originated from the middle layer mesoderm. Okay, so this is very important question. Now the fifth question will be retroperitoneal organs. Okay, what is mean by the retroperitoneal organs? Organs like kidney are covered by the parietal peritoneum only on their ventral side. Okay, see here, parietal peritoneum only on the ventral side. Peritoneum is nothing but a layer which is covering the kidney. Okay, so that's what parietal peritoneum on their ventral side. So these are all called as the retroperitoneal organs, as these parietal peritoneum which is present only in the ventral side, it is called as the retroperitoneal organs. Okay, now the sixth question will be, what will be the difference between exocrine and endocrine glands? Okay, so gland means they are going to secrete something. It may be the hormone. Okay, so as the hormones or any other secretion secreted by the gland, that has to be sent to the destined area. Okay, so whenever the hormones are carrying to the destined area, it will be carried to the ducts, carry through the tube-like structure called as the ducts. If the ducts are present, we are calling them as the exocrine glands. Okay, so if the ducts are absent, we are calling them as the endocrine glands. As there is no duct seen in the endocrine glands, we can call this particular gland as the ductless glands. Okay, so the exocrine gland, they are having a duct secreting the mucus, saliva, milk, etc. Okay, so they are the ducts. These exocrine glands, they are having a mucus, saliva, milk. All these were the secretions secreted by the glands. Okay, so example, salivary glands. So the endocrine glands, this is a ductless glands. Okay, so the ductless glands, they are also secreting the hormones. Okay, see here, exocrine glands are the glands which are they are secreting the mucus, saliva and milk. Okay, so endocrine are the glands, they are the one involving in secreting the hormones. So those hormones, all these hormones are not carried through the duct. So that is why it is ductless. They are the one carried by the blood. Okay, so say for an example, thyroid gland, they are secreting the thyroxine. Okay, now the distinguish between the holocrine and apocrine glands. Okay, so apocrine glands means the apical part of the cell, and this is the cell. Okay, and the apical part of the cell will be pinched off. Okay, so this is the pinched off. So this cell which is pinched off, so along with the pinching of the cell, there is also a secretion. Okay, so that is what a mammary glands. Okay, so the mammary glands will be, it is considered as an apocrine glands. Why? Because the apical part of the cell is getting pinched off along with the secretion. Okay, so now the holocrine glands. Here the entire cell disintegrates to release the contents. Okay, so to release the content, the entire cell has to be disintegrate. Not only the apical part is pinching off. Okay, so if the apical part of the cell is pinching off along with the secretion, we are calling them as the apocrine glands. Here, to disintegrate, to release the contents, the entire cell is getting disintegrating. That is what a holocrine. As holo means complete. The complete cell who is secreting is destroying. Okay, so that is what the sebaceous glands. Okay, so this is very important. Now, two substances secreted by the mast cells and their function. Okay, so what these mast cells secreted. Okay, and what is their function? So mast cell is going to secrete the heparin and histamine. Why? Because the mast cells having the granules inside the cell. Okay, granules inside the cell. Okay, so as these mast cells are having the granules, these granules are responsible for secreting this heparin and the histamine. Okay, so you should think that when we cut the hand accidentally, the blood will come out, and immediately after certain minutes the blood will coagulate. Okay, but the blood which is flowing inside the body, which is not coagulating, okay, which is not coagulating, which is not clotting. Okay, so that the prevention of blood clotting inside the body, that will be done by the heparin. Okay, which is considered as an anticoagulant. Okay, so now the another substance will be the histamine secreted by the mast cells. Okay, so this histamine is considered as a vasodilator. Okay, so the histamine is nothing but it is considered as a vasodilator, which is the one causing the inflammation. Vasodilator in the sense, consider this is a blood vessel. Okay, so this is a blood vessel. So this is a blood vessel, and if there will be an increasing blood flow, the blood vessel is very small, as the flow of blood is not coping up by this blood vessel. Okay, for that the blood vessel has to be elongated like this. Okay, so this is called the vasodilation, which is responsible for causing the inflammation. So this is done by the histamine. Now, the difference between the tendon and the ligament. Okay, so this is the this is the bone, and this bone is always surrounded by the muscle. Okay, and the bone and the muscle has been connected by the tendons. Okay, if the bone is connected with another bone, that will be done by the ligaments. Both ligament and tendon is nothing but a connective tissue. Okay, connection between something, either it will be the muscle or bone, or it will be the bone to bone. If it is, if it connects the muscle to the bone, it is called as a tendon. If it connects bone to the bone, it is called as a ligament. Okay, now the distinguish between the brown fat and the white fat. Okay, so white fat means it is found in the adults. Brown fat, it is found in the fetus and in the infants. Okay, so the white fat is single large lipid droplet, and it will be multiple small lipid droplets. So it is, white fat will be large and the brown fat will be small. Okay, so the white fat is metabolically inactive, and the brown fat here, it will be metabolically active, and this is the one. So whenever it is metabolically active, they are going to do certain functions. So as a result of all those reactions, all this metabolism, the energy is lost as heat. Okay, so that is what done by this brown fat. And the white fat, it is not metabolically it is metabolically active. Okay, so that's why it is not going to metabolically inactive. That is why it is not going to generate any heat. Okay, now the next question will be the strongest cartilage. What is the strongest cartilage? Where it is found? Okay, so the fibrous cartilage is the one strongest when compared to the elastic cartilage. All these cartilages, fibrous cartilage is considered as the strongest cartilage. Okay, so it is found in intervertebral disc and pubic symphysis. Okay, intervertebral disc, pubic symphysis. So this is all the thing which is where we can see the fibrous cartilage. Okay, so this fibrous cartilage is considered as the strongest cartilage. Okay. Now, The distinguish between osteoblast and the osteoclast. Osteoblast means this is the immature bone cell secrete the matrix and aids in mineralization. Osteoclast phagocytic cells involved in bone resorption. See here, osteoblast is the immature bone cell. The bone cells which are not completely mature is considered as an osteoblast. They are the one responsible for secreting the matrix and aids in mineralization. Okay. Now the osteoclast, which means the calcium, if it is accumulated, it is called as an osteoblast. If the calcium content in the osteoclast is removed, then we are calling them as the osteoclast. Okay. So this is what a phagocytic cells involving in bone resorption. The density of bone will be decreasing in osteoclast. If it is increasing as a result of mineralization, we are calling them as the osteoblast. Okay. Now, osteon. So, if the cell present in the kidney is called as a nephron. Okay. Nephron is a functional and structural function unit of kidney. And if it is a hepatic cell, the cell present in the liver is called as an hepatic cell. And if the cell present in the bone is called as an osteon. Okay. The Harvesian canal surrounding lamellae and lacunae form an osteon or the Harvesian system. So this Harvesian system we have discussed in the long answer questions, I think. Just go through it, okay. So how can we define the osteon? So it is nothing but a Harvesian canal. So if the Harvesian canal is surrounded by a lamellae and lacuna, we are calling them as an osteon, and this osteon is also called as the Harvesian system, which is in the bone. Okay. Now, the next question is Walkman's canal and what is its role? The Walkman's canal means transverse. It is transverse or oblique canals connecting the Harvesian canals or periosteum and bone marrow cavity. This is the one facilitating the communication and nutrient diffusion. See here, Walkman's canals, again it will be present, it is related to the bone. It is transverse and oblique canals. Again, we have discussed this Walkman canals whenever we discussing the long answer questions, I think. Okay. So, the canal which is connecting the Harvesian canal, periosteum and marrow cavity is called as a Walkman's canal. Okay. So, it is a transverse oblique canals, and why it has to connect with the Harvesian canals and periosteum and marrow cavity means to facilitate communication and to transfer the and to diffuse the nutrient. Okay. So the nutrient will be supplied with the help of the Walkman canals as it is associated, connected with the Harvesian system. Okay. Now, what is meant by the sesamoid bone? The sesamoid bone, it is formed by the ossification in tendons. Okay. Ossification in tendons, which means the bone is getting ossified. Okay. So that is the sesamoid bone is formed by the ossification in tendons. So just now we have studied what is meant by tendon. So it is a connecting tissue which is connecting the muscle to the bone. Okay. So how this tendon, how this sesamoid bone is formed means by the ossification of this tendon. So that as a result of this ossification we can form the sesamoid bone. Okay. So the example will be the patella. Okay. So the patella, which is the kneecap. Okay. We can see the sesamoid bone in the kneecap. Right. Now, what is lymph? How it is differ from the plasma? Okay. So what is the difference between lymph and the plasma means? So we have discussed this term when we have discussed about the discussed in the long answer questions. Okay. Body fluids and circulation. See, if the blood without RBC is lymph. Okay, blood without RBC is lymph. Okay, see here. A lymph, it is a colorless fluid. Okay, lymph is a colorless fluid as it have no clotting factors. Okay, as it have no fibrinogen, if it is colorless in nature. Again, it lacks RBC, it lacks platelets, it lacks large plasma proteins. So all these are lacking in the lymph. Okay. So has more leukocytes than plasma. When compared to the plasma, lymph has more leukocytes. So these are all the major difference between the lymph and the plasma. Okay. Now, what is the hematocrit value? Hematocrit value. So see here, the percentage of total blood volume which is occupied by these RBCs, the total volume of blood. See here, we are having a total volume of blood. So in the blood we are having RBC, WBC and platelets together with all other proteins also. Okay. So when compared to the total volume of the blood, the place which are occupied, the volume which are occupied by the RBC. Okay. So that is what the hematocrit value. Okay. Hematocrit value means the percentage of the RBC volume which is present in the total blood volume. Okay. So that is what the hematocrit value. Right. Now, what are this intercalated disc and what is its significance? Okay. The intercalated disc means the dark lines across the cardiac muscle. The cardiac muscle have some dark lines. So the dark lines which is present in the cardiac muscle, and the cardiac muscle will be present only in one organ, that is nothing but a heart. Okay. So the heart, they are having a cardiac muscle. So in the muscle they are having a dark lines. So these dark lines is considered as the intercalated disc. Okay. So this intercalated disc act as a boosters of muscle contraction. Okay. So the boosters of muscle contraction, because see here we know that the cardiac muscle have the capacity to contract and relax. Okay. Now this cardiac muscle acquires the property of contraction. How? Because of the presence of the intercalated disc. Now the cardiac muscle is highly resistant to fatigue. Okay. See here, it is highly resistant to fatigue. Justify. Due to numerous sarcosomes, myoglobin and rich of blood supply, it supports continuous aerobic respiration. Cardiac muscle highly resistant to fatigue. See here, we often come across with the word muscle fatigue. Okay, so this muscle fatigue in the sense, as the muscle cells are devoid of oxygen supply, we can call them as a muscle fatigue. Okay, but here there is a speciality of this cardiac muscle. They have a high resistance to fatigue due to numerous sarcosome and myoglobin. So if the hemoglobin present in the muscle cell, we are calling them as the myoglobin, and they are having a constant rich supply of blood. Okay, then how come the cardiac muscle will experiencing the fatigue? Okay, so all these structures supporting the continuous aerobic respiration. So this is important thing. Now the next question will be the distinguish between the nucleus and ganglion in the nervous system. So what will be the difference between the nucleus and ganglion? The group of cell bodies present in the CNS. If the group of cell bodies which is present in the central nervous system, we are calling them as the nucleus. If the cell body is present in the peripheral nervous system, we are calling them as a ganglion. See here, both the word nucleus and the ganglion are similar. Okay, based on the location, we are calling with two different terms. Okay, if the cell body present in the central nervous system, we are calling them as a nucleus. If it is present in the peripheral nervous system, we are calling them as a ganglion. So this is the basic thing. Now, the distinguish between the tracts and the nerves in the nervous system. What will be the tracts and nerves? Both are same. Again, see, if the axon present in the central nervous system, we are calling them as a tracts. Group of axon, if it is present in the central nervous system, it is considered as a tracts. If the group of axon present in the peripheral nervous system, we are calling them as the peripheral nerves. That is nerves. Okay. Now, distinguish between the white matter and grey matter of the central nervous system. Okay. See here, the white matter, it is contained in the myelinated nerve fibers. If the axon has myelin sheath and the nerve fibers pass through it, we are calling them as a myelinated nerve fibers, and that causes. And this white matter contains the myelinated nerve fibers, and the grey matter, they contains the non-myelinated nerve fibers. This is what a grey matter. Okay. Now, what are microglia, origin and function? Okay. See here, microglia means phagocytic cells of mesodermal origin. Okay. See, there is an outer layer, middle layer and inner layer. Okay. And the middle layer, this is called as a see here, the phagocytic cells of mesodermal origin, we are calling them as the microglia. See, so we are having 5 different types of cells in WBC. Leukocytes, neutrophils, basophils, eosinophils, lymphocytes, monocytes. All these were the 5 different types of cells in the WBC. Okay. So, monocytes. If the monocytes, if it is streaming in the blood, we are calling them as monocytes. If it is reaching certain tissue, they develop some structure, and we are calling them as a macrophages. And this macrophages is the one doing the process of phagocytosis. Phagocytosis means engulfing the cells. Okay, that is what a phagocytosis. So, if the macrophages, if it is present in the lungs, we are calling them as the alveolar macrophages. If it is present in the kidney, we are calling them as the some Kupffer cells. If it is present in the brain, we are calling them as the microglial cells. Okay. See, so this microglia is nothing but a macrophages, which is a mesodermal origin, and this is a phagocytic cells. Clear? Now, it becomes active during infection or injury in the nervous system. Okay, so whenever it is active, when there is any infection or injury in the nervous system, this microglia will be active. Okay, other than that, it will be inactive. So this is an important thing, just keep in mind, right? Now, so that's all about the short and very shorts in the structural organization animals. Now comes the very shorts in the breathing and exchange of gases. Okay. Now, the first question will be define the vital capacity and what is its significance. Vital capacity means the maximum volume of air a person can breathe in after forced expiration. Okay. Now I am inhaling. Okay. Now I am forcefully exhaling the air. Forcefully expire all the air. Exhaling all the air. Now, after the forceful expiration, the maximum volume of air the person is inhaling. Okay. The maximum volume of air a person can breathe. That is called as a vital capacity. Okay. Now, it includes the tidal volume. It includes the tidal volume, inspiratory reserve volume and expiratory reserve volume. Okay. So, the tidal volume, which means the normal inspiration, normal expiration. Inspiratory reserve volume and expiratory reserve volume. All these together we are calling them as the vital capacity. Just keep in mind, this is an important thing. And see here, what are conchae? Okay, conchae means it is also called as turbinals. Okay, conchae, it is also called as a turbinals or 3 thin twisted bony plates present in the respiratory part of the nasal chamber. Okay, see here. Conchae is nothing but a terminals which is present in the nasal chamber. Okay. And this conchae means it is 3 thin. Okay, it is 3 thin twisted bony plates. This is all their structure. How can we define their structure? Means it is 3 thin twisted bony plates and it is present in the respiratory part of the nasal chamber. Okay, so this why this conchae is present in the nasal chamber? Just to increase the surface area. Helps in warming. So whenever we are inhaling the air, it will be chill. Okay, because the environmental the temperature which is present in the atmosphere decide the humidity of the air which is present in the atmosphere. Whatever the temperature of the air we are inhaling, whenever it is came out, it will be very hot. Okay, and that will be done by this conchae. Okay, so they are increasing the surface area. Not only this, they are helps in warming, moistening and filtering the inhaled air. All these are called as a all these were the function of the conchae and their structure. Right now. The next question will be chloride shift. Very important. See here, the chloride shift will be done in the blood to maintain their pH. Okay. Yes, this chloride shift refers to the movement of chloride ions. See the movement of chloride ion, that is Cl plus into the RBC. This is the RBC. Okay. So the chloride ions will be entering into the RBC when the bicarbonate ions move out in the plasma. Consider this is a plasma. This is the plasma, and if the Cl minus is coming out, the HCO3 minus will enter into the plasma. Okay. This is what a chloride shift. Okay. See, this why it has to move, why the chloride has to came out and the bicarbonate has to came in. Why the bicarbonate has to came out and the chloride has to came in means to maintain the buffer, to maintain the pH. Okay, so that is why the pH of the blood will be basic, slightly basic, it is neutral. Okay, so this maintain the ionic balance during the carbon dioxide transport in the blood. Okay, so this is very important, right? Now, name the muscles that helps in normal breathing movements. Normal breathing movements, the main important muscle will be the diaphragm and the external intercostal muscles. Okay, diaphragm and the external intercostal muscles. They are the one primarily involved in normal breathing movements. Okay, this helps in expanding, contracting the thoracic cavity during inhalation and exhalation. Whatever, either it will be exhalation or internal inhalation. The diaphragm, external intercostal muscles and internal intercostal muscles. All these muscles are involving in the normal breathing movements. Okay, so that's all about the very shorts in 2 chapters. So we have completed 4 chapters, very short answer question. We'll meet you in the next class with another chapter, very shorts. Okay, thank you so much.